last 3 Thursdays at this time, we're going to have Prof. Tad, who's very kindly offered to do a bit of NGS related bioinformatics with you. And remember that this will count 10%. The questions in that he gives you will count 10% of your module. And that class participation mark, if you can tell me who were 1 or 2 key means that really joined in, it still counts here. So, cool. And remember, no lecture tomorrow, but then we'll have 4 lectures in total next week.
All right. Well, I'm going to swing this around. At this point, just hopefully it stays aimed. Am I in shot at all? Oh, that's good. Hi there. I'm Dave Tapp. I am a researcher over at Stellenbosch University, but I'm here on Medical Research Council funding. And that means that I get to work with you guys too, because they really want to make sure everyone in town gets the opportunity to get more bioinformatics in their training. So what is a bioinformaticist? I was trained at the undergraduate level as a biologist. I did a biology undergraduate degree at the University of Arkansas. Then after that I did a PhD in molecular biotechnology at the University of Washington. Neither of those degrees is ostensibly bioinformatics, but that's true for most people in bioinformatics. We have people transfer in from the world of physics, from the world of statistics, from the world of biology, as you might imagine. And all of us find some way to maximize the amount of information that we can get from biological data. How that takes effect in each person's research is going to be different. But for me, the focus has been on discovering protein identities based on tandem mass spectrometry. So the folks over in the Mass Spec Center are the ones I work with most frequently over here at UWC. But I was really glad to get the chance to talk about bioinformatics as it touches next-gen sequencing. Now, the term next-generation sequencing has already been out of date for several years, right? So you might instead think of it as massively parallel sequencing. And that really captures the nub of it, that modern sequences are not better because they produce longer sequences or because they produce sequences of greater quality, but rather because they can sequence millions of templates all at the same time. So this massively parallel sequencing is the chief advantage that we get from NGS. So today we're going to talk about kind of the first set of challenges that we think about when we're working with next-gen sequencing. Now we can sequence a new genome for an organism in a rather routine experiment. But there's a really, really big difference between a bazillion reads and a very, very long sequence that we think of as the assembly of that genome. There's yet another step of annotating that. So instead of having just a whole bunch of letters, we have annotation that tells us, well, genes are here. We think this is a protein coding gene right here. We think it produces this product, and this is what that product does. So today we're going to talk about how we go from a whole bunch of sequencing reads to contigs to an annotated genome. So that's the nub of today. So we're going to sort of split this into 2 pieces. Think of this first one as from reads to chromosomal assemblies. And then the second stage of this as the annotation, how we figure out what those letters actually represent in terms of biological modules of activity. So along the way, we're going to talk about why Pairedend sequencing is so highly valuable in the process of assembly. We're going to talk about repeats as and what do we do with with all these remnants in our genome that are not terribly functional DNA at this point. And finally, we're going to talk about Kmers. A Kmer is a really important concept that shows up in assembly and a lot of other topics as well. So I want to make sure you have some notion of what that means when you hear the term. Then we're going to discuss annotation. Think of this as the process of translating sequence to known biological entities. We're going to talk about the magic of hidden Markov models. They're a fairly scary concept, but I think that you at least need to know what you can expect from one when you encounter it. And from there we'll talk about homology search, including what a lot of people think of as the killer app of bioinformatics. And finally, we'll talk about domain and motif matching to help us determine what a protein of unknown homology does. So we'll walk through that bit by bit. It's a lot of material. I hope we can do it all in an hour. If it runs to 90 minutes, is anyone going to boot me out? Okay. This will all be stuck on YouTube as well, so feel free to write down the stuff that isn't on the slides. You'll have access to the slides yourselves very shortly. Okay, so there are a lot of people who get the impression that bioinformatics is just this one thing, right? They have maybe one narrow silo of this that they think about. In this case, I'm showing many of the active research fields that take place within the sequencing world in particular. Down the side, we have all these green boxes that represent a lot of the things that we tackle in gene expression. How do genes, which genes are on or off under certain circumstances? We'll talk about that a lot more in 2 weeks. A week from today, we're going to be talking about amplicon sequencing bioinformatics. And right now, that doesn't really appear here. So we'll have a whole other set of topics that throw into this. Now, is this all of bioinformatics? No. Because I've already shown that my title down there at the bottom says it's for sequencing bioinformatics. And I work in an altogether different area of bioinformatics, with the world of the proteome. So be aware that bioinformatics is going to span much, much bigger than what I can cover in just 3 lectures. All right, on we go. So Pairedend sequencing. Hopefully you've had some exposure to this concept already. We have one template, one insert sequence in this case, and we're going to get a little patch of sequence from one end of it and a little patch of sequence from the other end of it. That is highly valuable to us because these 2 sequences we know are not independent of each other. We know that they are opposite in orientation, and we know that they're relatively close to each other. With a lot of sequencing, we don't even know exactly how close they are. We may know that they are, say, within 2,000 base pairs of each other, or we might know that they could be as little as 500 base pairs away from each other. We don't really know that with a lot of the ways that we produce our inserts. But we do know that their orientation is opposite, and that they came from one piece of DNA, pointing inward. So, how is that useful? Now this comes from a paper from Jared Roach, a friend of mine from grad school. You never know who it is in your very own graduate class who's going to go on to do something amazing. And it turns out that Jared was a very, very sharp cookie even back in grad school. He wrote this key paper that demonstrated why having paired end sequences greatly improved our ability to produce successful assemblies. Now I'm throwing around a lot of terms here, so let's try to be clear on what we mean by them. We have a read. A read here represents one contiguous stretch of sequence letters produced by the sequencer in a single experiment. We have a process of assembly, which is trying to figure out which reads align with each other, which allows us to get longer stretches of DNA we call contigs. So if we are starting with reads as our input, the process of assembly is going to produce contigs from those. So in this case, we have a whole bunch of different bits of DNA that gave us paired ends. So we see those light blue stretches of sequence with dark blue patches on either end. Those represent our inserts with a read on either end. And we see that some of those blue patches, the dark blue patches, stack up on top of each other. And that's really grand because then we can simply detect those overlaps and from those overlaps deduce a longer stretch of sequence. But on occasion, we're going to have some little holes in that assembly. We're going to have places where we don't have any dark blue patches, but we may have a light blue region that stretches through there with patches on either end that have been sequenced, but this particular area gets 0 coverage. That's going to happen by random chance, because in almost every case, the sequencing that we do today is based on random library creation. So how are we going to figure out that these contigs actually belong next to each other? This is where paired end sequences show their worth. All right, now I'm going to start by mentioning that our own personal genomic evolution has come through some patchy times in the past. I believe around the time that primates separated from other apes, we had this kind of war going on within our genomes. And we have a lot of extra DNA within our genome that are evidence from this sort of genomic war that was going on. So the ALU sequences are a prime example of this. ALU sequences are these sort of, they're a type of short interspersed repeat element that appears in bazillions of copies throughout our genomes. So when we talk about only 1% of our DNA actually codes for proteins, some of the rest of that 99% reflects this kind of evolution of pseudogenes and so on through our, these are regions of DNA that are able to produce new copies of themselves over time. Our genomes have found ways to defend themselves against this, the rapid spread of these repetitive sequences. But a lot of the mess in our genome comes from, and a lot of the variation in our genome has come from these repeats that have been spread throughout. So this is a brief taxonomy of all those different extra segments that got thrown into our genomes over time. And not all of them are this kind that can create new copies of themselves. Like mini-satellite and microsatellite DNA are mostly just repetitive sequences. They're not necessarily, those aren't transposons in that they can insert themselves at new places within our genomes. But we have bazillions of repeats like this in our genome, and that is a real challenge to sequencing. Okay, so now let's get right to the nub of assembly. Here we have a problem where repeats can cause things that are quite far apart in the genome to look like they're quite close together. So up at the top of the box A there, here we're working from a paper published back in 2002 from Batzoglu. He was publishing his assembler algorithm called Arachne. And I want to point out that was this fellow's PhD thesis, just turned into a paper. This person's PhD project created an assembler which was the first to be able to assemble the fruit fly genome on a desktop computer. It's kind of an astonishing capability to have. But the ideas in the Arachne assembly have been used all over the place. So let's look all the way up here at the top of box A. Imagine that somewhere in the genome, you have a gene A and a gene B here, and they're separated by a repeat. At another point in the genome, somewhere far away, we have a gene C and a gene D, and these are separated by the same repeat. So we see that, as I mentioned, these repeats are notable because you have lots of copies of them throughout your genome, which means that even though repeat 1 and repeat 2 are in different locations in the genome, their sequences are nearly identical. So the problem that you get is that when you do a sequencing, what does that mean? No, somebody else says, okay. When we have a sequencing read, it might be that you read a little bit from gene A into the repeat, and that's all the read contains. And then over at the right, we have another sequencing read, here labeled with that little Y, that starts in the repeat and runs into gene D. If all you have are the sequences of X and Y, you might erroneously think that because they both run through the repeat, that gene A is next to gene D, separated by the repeat. Do you see the problem? So repeats are really, really troublesome to deal with in the context of this. So we need to be able to screen out those regions of sequence that represent repeats. That means that we're going to have contigs that are broken at repeats, in essence, but hopefully we have other large-scale means of deciding where these contigs fit together. All right, next, sequence correction. Imagine that you have 5 reads that all pass through the same region, and you can see in this position near the end of that alignment of them, that we have C appearing in 4 of the reads and T appearing in 1 of the positions. Sequencers make mistakes, right? So it's possible that that these are all reads from exactly the same region that have this correction needed. So what we also gain along with these sequence reads is some measure of quality for each letter in that. Have you guys talked about Fred scores at all? Ah, okay. Well, I'm going to help you interpret these then. So we have, in the top sequence, we have a score of 20 given for this letter. We have a score of 35 in the second. In the third, we have a score of 30 given to a T, 35 for C, 40 for C. I want you to think of this as a log base 10 scoring system. And the higher the number, the lower the probability of error for this letter at this position in the sequencing read. So when you see a score of 20, the first rule is move the decimal point, so call that a 2 instead of a 20. Raise 10 to that power and put 1 over it. So the 20 becomes a 1 in 100 chance of a miscalled letter. Now that 30 is a little more problematic. Remember, a higher score represents a greater confidence at this position. So the score of 30 here represents a 1 in 1,000 chance of error. And a 40 represents a 1 in 10,000 chance of error. Have I got this straight? Yeah, 30 was 1,000. Yeah, so basically, this is kind of a logarithmic thing. 10 to the minus 2 is 1 in 100, 10 to the minus 3 is 1 in 1,000, 10 to the minus 4 is 1 in 10,000. So the sequencer is not just saying this letter appears here, it's saying this is how sure I am that this letter is correct. So when we do sequence pileup, when we do this assembly process, we need to have some kind of voting system that places greater weight on letters that we have very low chances of error on, and allows us to recognize things like this T that are faulty. Now the solution that Batziglo put together was that when you have a letter that's badly outvoted here, remember that 40 is 10 times as confident as the 30 for that T. His solution is that we replace the T with a C and give it a score of 0, essentially saying that this can't add to our confidence that C is correct at this position, but we can scratch this one out and effectively say it's been outvoted at this position. Okay, so I want you to be able to translate numbers like these into probabilities if at all possible, because you see logarithmic scoring systems used all over the place in bioinformatics. And so when you see a 40 or a 30 or a 20 on Fred, I want you to be able to keep in mind which of those is most confident. I haven't written my questions for the test yet, but we'll see what that looks like, right? Normally I would do a whole lecture on Fred, but we haven't got time for that. Okay, now, I mentioned Pairedend reads were going to be darned essential, and here's where they show up. So we have contigs that have been formed by these contiguous, comes from the word contiguous, from these contiguous regions of sequence that we have complete sequence for, all the way from one end to the other. But we've got these little patches where we don't have sequences just by random chance. And we need to be able to figure out how to patch together these bazillion patches of these bazillion contigs we've produced from our sequencing reads. Now, before, we saw a light blue line representing an insert sequence with a dark blue patch on either end to represent the parts we actually sequenced, the paired end reads. Here, it's shown as an arc. A single insert sequence is shown as an arc, and we have a little patch at either end. That represents the sequence read at either end of that insert. And as you can see, even though we don't have any sequence between these contigs, we do know that several inserts bridge this gap between contigs. Remember, if we have paired sequences that were produced from the same insert, we know their alignment versus each other. We know that this one is here and this one is here, and that something connects them. We may not know how big a space that is, but we do know that they're adjacent. So when you have a very large number of contigs, you can still figure out how they tile together based on the fact that inserts bridge from one to the next contig. All good so far? Okay, feel free to ask questions in flight. Your voice will show up on the recording, but if that doesn't bother you, feel free to ask your question. Okay, we're going to move ahead. So, I mentioned that Kmers are a really useful concept to know. And this is the sort of thing I'd probably ask a question about, honestly. So, why do we need to know Kmers? I'm going to show a section of text soon that explains the advantages of a Eulerian path approach versus a Hamiltonian cycle approach. You don't need to know that as much, only to know they exist and what kind of field they fall into, really. But Kmers is a very valuable thing for you to know about. So how long a stretch of sequence read do we get from one of today's sequencers? They produce really long reads. Okay, people are a little unsure on this one. And there's a reason for that. Although we think of the dominant sequencers we use every day to produce very short reads, like about 100 or about 200 nucleotides, there are sequencers available today, like those I think from PacBio, I think, that produce very long reads, thousands of nucleotides. So when I talk about today's sequencers versus yesterday's sequencers, I'm usually talking about something like capillary electrophoresis instruments that were used in the late 1990s to complete the Genome Project. They produced, at best, about 600 nucleotides in a given sequencing experiment. The Illumina sequencers that we find everywhere today generally produce sequences that are around 100 or 200 nucleotides in length. Okay? So I think of modern sequencers, as opposed to the old school Sanger sequencers, as producing shorter sequences in general. That generalization is not bulletproof, though. Okay, so let us imagine for a moment that we're dealing with a sequencer that outputs reads of about 100 nucleotides in length. How many different 25 nucleotide sequences can I pull from that 100 base pair stretch, that 100 nucleotide stretch? I hear 4. Everyone on board with that? Okay, so what that asserts is that from this 100 base pair stretch, we can put a 25 mer here, another 25 mer here, another 25 mer here, and another 25 mer here. That's 4. But that's not how Kmers work. Instead, we ask, how many different 25 mers can we pull from this, moving one letter each time? Okay, so our first 25 mer comes from one end of the sequence, and then we scoot by one nucleotide, and we have another Kmer, and we scoot by one nucleotide, and we have another Kmer. All right, so now with that in mind, how many different 25 letter Kmers can we produce from a single 100 base pair read?
100.
75.
I like this. Good discussion. All right, so I heard some people saying, I heard 100. I heard 75.
75.
I'm hearing strong votes for 75. That's actually really close. It's really close. I believe the answer is 76, because you have the one more position from. But yeah, you have this moving window that starts at residues 1 through 25, 2 through 26, 3 through 27, and so on. Keeps scooting it. And I believe that you have the one position, and then you can move this last position to 75 other positions. So I think the total is 76. If my math could be wrong there. I was thinking about this problem. Was I sure I knew the answer to that question when I asked it? Okay, so think about it that way. We're going to start with a read, and we're now going to extrapolate all of these different 25 mers from it. When we say Kmer, we're just saying that we don't necessarily use the value 25. Maybe we're trying to produce 28 mers or 20 mers or something like that for a particular application. 25 is a pretty common number that we see in reference to nucleotide sequence. So we may start with 100 mer, that is our read, and from that we're going to blow out 76 25 mers from it. Now, this is the crazy thing. We can do this for all of the reads produced in an instrument. Now you have a MiSeq here. What kind of sequencer do you use here? MiSeq? All right. So how many different reads do you get from a MiSeq in one experiment? It's a lot. It's a lot. Okay. Can I use the number 1,000,000? Am I way off with that? Higher or lower? Want to go lower? All right, we're going to use 1,000,000. We're going to use 1,000,000. All right, I actually don't know the specs on the MiSeqs, but that's okay. So let us imagine that we have 1,000,000 sequencing reads, each of which is 100 nucleotides in length. we're now going to blow that out 76-fold into Kmers space. So whenever you see a computer scientist doing something like this, you'd better be asking, why would you make that problem worse? And I work in the field of proteomics. One of the things that I'm always thinking about is, we identify peptides. So that one of the first things we do is make our problem worse. We started with proteins, we digest them up with peptides. We've now made more peptides to identify than we started with in the world of proteins. So you'd better have a good question in your mind every time you see someone doing like that. Why would you make the problem worse? The answer is that we want to determine which reads intersect with other reads. So you can imagine that we could do this the brute force way. My computer science professor always used to call this the brute force and ignorance approach. That you start with a given read, and then you compare it to every other read to see if they overlap. You can do that, right? So you can ask, how many different pairwise comparisons do I have to do, starting with 1 million reads, to look for these overlaps? Do you guys know the answer to that one? This is called the cocktail party problem, another thing I love talking about. If you've got 20 people at a party, how many different handshakes can you have? It's the same problem. So the answer here is that you don't need to compare a sequence to itself, and if you've compared A to B, you've also compared B to A. So the answer becomes n times n minus 1 divided by 2. So if you have 1 million sequences and you want to compare every sequence to every other sequence, it's going to be something on the order of 500,000 times 999,999. It's a lot of comparisons. It's bad. On the other hand, we can sort like demons in the world of informatics. So what we're going to do is build out the set of all 25 Kmers to be found in any read, sort them, and then we determine overlaps by shared Kmer content. Think about that. I have 100 base pair read over here. I have 100 base pair read over here. We know that these contain a large number of shared Kmers. Therefore, we know they overlap. Okay? So that is the key to how we make assemblies from reads. There are some limits here. Imagine that you had 200 base pair reads that overlapped by 10 nucleotides. If your Kmers are 25 nucleotides in length, you will not detect an overlap between these reads. Everyone sees why? Because they have no shared 25 Kmers. They have a shared 10 Kmer, but not a 25 Kmer. Similarly, if you've got base calling errors, they're going to foul things up too. Because you're going to see cases in which you have a huge number of shared Kmers, except for this particular base calling error set that splits out to different Kmers. Okay. Oh, yes, there's a question.
So when you do your runs or when you're doing your assembly, how do you select for the number of Kmers that you're going to use?
That's a good question. How do you figure out the correct Kmer length for doing your assembly? Generally speaking, that we don't fiddle with that parameter a lot. All bioinformatics software has lots of parameters that can possibly be changed that will alter the final outcome. Part of this relates to the kind of alignments you're trying to perform. In this case, we're trying to do nucleotide to nucleotide alignment. If you set Kmer length if you set K to 6, for example, there are not that many possible 6-letter nucleotide sequences. There's only 4 different possibilities at each location. So generally speaking, if you're doing an alignment in protein space, where you have 20 different monomers rather than the 4 different monomers you have in nucleotide, you can get by with a shorter K. But when you move into the world of DNA alignments with only 4 at each position, 25 is pretty standard. You can cut that down to 20 and you can increase it up to 30, something like that. You won't see huge differences in performance, but it will change. Good question. Other questions before we move ahead? All right, now let's read a slide Oh, sorry. This is the friendly figure that shows how this information gets used on the very, very large scale. So your pronunciation of De Bruyne is probably a whole lot better than mine because my Afrikaans is nearly non-existent. But the De Bruyne graph represents a way to link all of these Kmers together. In fact, we digest this problem down a lot worse and start thinking about all of the prefixes that you can draw from a particular Kmer and all of the suffixes that you can draw from a particular Kmer, which is to say all of the sequences starting at the beginning of the Kmer and all of the sequences that end at a Kmer. But we want to find ways to link together these different Kmers to allow us to walk through the entire alignment. So this is the De Bruyne graph is one of these elements that we use for assisting us in walking through the sequence. Now, in case your mind is not already blown by the problems of trying to work through these Kmer stacks, I want to just point out that modern short-read assembly algorithms construct a De Bruyne graph by representing all Kmer prefixes and suffixes as nodes, which is to say all the sequences at the start of a Kmer, all the sequences that end at the Kmer boundary, and then drawing edges that represent Kmers having a particular prefix and suffix. For example, the Kmer edge ATG has a prefix AT and a suffix TG. People can see that part, right? Because AT starts at the beginning of the Kmer and it runs 2 letters in, and TG ends at the last two. Finding a Eulerian cycle, this is the standard method that we use for threading through these graphs, allows one to reconstruct the genome by forming an alignment in which each successive Kmer from successive edges is shifted by 1 position. This generates the same cyclic genome sequence without performing the computationally expensive task of finding a Hamiltonian cycle. So 6 years ago, this was published. This method has become very, very widely used. I feel that it's so complex in computation I wouldn't try to explain it, but this URL is very good if you want to understand better how we can trace through all of these wild Kmer graphs to get a contig. Okay. So let us imagine then that what we've accomplished in the first half of this lecture then is to start with 1 million reads. We decided there were 1 million, right?
Yes.
And produce a very long sequence from that. Okay, so having produced one very long sequence from our sequencing experiment, have we produced a genome? Okay, that's a very valuable thing. So we have produced a long sequence, but the problem of annotation still remains. Assembly got us from reads to this point, the very long sequence. But now we want to figure out what points within this very long sequence are ones we actually care about. So for that, we're going to talk about the problem of finding genes by hidden Markov models, one of the very popular ways of doing it. Recognizing orthologs by sequence alignments. In sequence alignments, you can find whole semester-long classes just on the subject of alignments. So we're just going to touch it very lightly and move ahead. We're going to talk also about assessing function. How do we figure out what a sequence actually does? Okay, so hidden Markov models. I remember when I first started teaching bioinformatics classes about 10 years ago, I was really concerned that I was going to have to teach hidden Markov models because I found them bewildering and baffling. I still find them a little bewildering and baffling, frankly, but that's okay. I think a lot of people do. When you have a biological feature that you want to find, like a protein coding genes, you can break up that feature into its component parts and figure out what kind of sequences are most likely to be seen for each of those parts. When we translate that into hidden Markov model language, we have these different parts of this biological entity we're trying to find. Those are all called states. Okay? So those states are the substructures of this large thing. We often talk about hidden and visible when we're talking about hidden Markov models. The states are considered hidden. We infer from this sequence that it is a protein coding gene and it has these little parts to it. So those parts are hidden from us. What we can observe is sequence what we want to infer are the states that describe how they relate to each other. So the emission probability says that when we're in a particular state, what are we most likely to observe from this? So presumably, you guys have heard of the term promoter. Promoter. Okay. So what is a common sequence emitted from a promoter? What kind of sequences do you see in a promoter? Does anyone know? All right, so I'm hearing ATG and start codon. That is not a promoter, actually. Right. The promoter is upstream of the protein coding part. Has anyone heard the name Tata box? Ah, we did have a Tata. Okay, great. All right. The Tata box is a common form of promoter. So if you're in the promoter part of a region of a sequence, you're likely to have higher probabilities for T and A residues within the read. Okay. So the emission probabilities in a promoter region are key to producing lots of T and A letters on the sequence. That's not a phrase I want to say too quickly, sorry. Okay, so then we get to transition probabilities. So we have these different pieces within a gene that relate to each other in some way. If you are in an intron, the next thing that you're going to hit is an intron-exon boundary, and then you'll hit an exon. So the transition probabilities talk about this stepping from one state to another as we walk across this structure. And we have algorithms that allow us to teach our model what these probabilities are from known examples. So I have some set of sequences that I know are proteins. I know which parts are intron-exon boundaries. I know where the start codon is. I know where the promoter is, etc. And I can say, this is how my model fits to this sequence. So learn the probabilities for emissions and learn the probabilities for transitions from that. Then go find more like this. You see that? So we have algorithms for learning the probabilities of the model. We have algorithms for seeking out other examples of it in an unknown sequence. Okay, so what does that look like? So here I have an example of gene scan. I'm showing you half of the model because the other half is looking for protein coding genes on the other strand. So it's just symmetric like that. So we have intergenic regions. We have promoters. We have a 5' UTR. Does anyone remember what UTR stands for?
Urinary tract.
Okay, you got that. Great. So untranslated regions. It's not untranscribed, it's untranslated, right? Okay, good. I got to remember this stuff. You might have a very simple gene in which you have an exon. Oh, thanks. And then you have a 3' untranslated region, a poly A signal, and then you're right back in intergenic space. So something like that is a really simple model through this. But let's imagine a more complex model where we have exons and introns that are alternating in space. So here we have our promoter, we have our untranslated region, we have an initial exon, we have an intron, and then we can step from there to another exon, and then back and forth. Now I've had to spell out 3 different kinds of internal exons here, because I've got FR0, FR1, and FR2. Those are reading frames. So it may be that you have an intron-exon boundary that falls in frame 0, or in frame 1, or in frame 2. And so when you step back into the exon, you have to remember your reading frame that you started that intron with. Okay, so after you get through that shuffle, you finally step into an exon final, a 3' UTR, a poly A sequence, and then the intergenic region again. So a model like this, even as complex as this, can be trained on known genes, and from it, go find more that look like it. Now, I would observe that these models tend to be a little bit prone to false positives. So you may have pseudogenes in a genome that look a lot like the real thing, that are dysfunctional, that get picked up as positives on GeneScan. and that we have to sort of look at with fresh eyes to recognize is actually a false positive hit. Okay. That's jazzy, I like that. Cool ringtone. All right, let's move to the next slide. Okay. So, having found a long stretch of DNA, we've now called these areas as protein coding regions. That's great. Next, we need to figure out what it does. And far and away, the easiest way to determine the function of a particular protein coding gene is to align it with known coding genes. So, we have sequenced a lot of different species out there. If I have sequenced the house cat, for example, what might I compare it to in order to determine what different parts of it do? Yeah, I might have another cat that's been sequenced, maybe a lion, for example. So I could leverage the lion to recognize the house cat. There's actually another thing I'm not talking about here called synteny that we can use. Synteny, S-Y-N-T-E-N-Y. If you have multiple species that you've sequenced and annotated their genomes, if you have gene A, gene B, gene C in a lion in order on a chromosome, you might expect that in the house cat, you also have gene A, gene B, gene C, the orthologs. You see that? Okay, so the first reason that we want to align sequences is that we may have orthologs, the same gene in a different species, to look for. We also want to recognize paralogs. A lot of times our genomes have duplicated genes over the course of our evolutionary history that we can, where for example, you made a copy of a particular area of DNA in the genome, and now one of them does X and the other one does Y. That's possible. Okay, so recognizing paralogs is also valuable. And of course, recognizing conserved regions. If you have a great many sequences from a great many different species, and you want to know which parts of this sequence are most essential to carrying out this function, recognizing those where mutation is not tolerated is very, very powerful. So all of those are good reasons to align sequences. What's our time check? Are we sitting at an hour?
45 minutes.
40. Oh, good. We're moving right along. This is good. All right. So from a very high level, when we look at alignment algorithms, we want to ask, are we looking at global aligners or local aligners? So if you're looking at a global alignment, this is something that's going to try to match every part of this sequence to every part of another sequence. So algorithms like Smith-Waterman and Needleman-Wunsch are examples of those. Smith-Waterman is actually a local aligner, my bad. The original Needleman-Wunsch algorithm is a full-length sequence aligner, a global aligner. Local is a lot better if you're trying to recognize particular shared features within the sequence, rather than trying to match the totality of this to the totality of that. Okay, the next, optimal versus heuristic. You will see that a lot of the original algorithms that were published for alignment were designed to produce what was provably the best alignment out of an entire database. So things like Needleman-Wunsch and Smith-Waterman are examples of this. However, what we've seen is that a lot of people are willing to deal with results that are not provably the best, but are still very good. And the banner example of this is BLAST, or the algorithm FASTA for that matter. So BLAST is a very high-speed algorithm that, although it will not give you the demonstrably, provably best match from a database, will still give you very high-quality matches and do it in a fraction of the time required for something like Smith-Waterman. So optimal versus heuristic is a good distinction to remember. Evolutionary distance. If you are looking at sequences that are very similar to each other, like house cats and lions, as we mentioned, you can probably do so on the basis of DNA, and you will detect differences that way that are relevant over very small evolutionary distances. However, if you are looking at something like yeast and human, we share an ancestor with yeast, but it's pretty far back at this point. You can do that using protein to still be able to capture signal over such a distant evolutionary space. So I'm not really going to spend much time, any time in fact, talking about multiple sequence alignment, but if you're using multiple sequence alignment over long evolutionary spaces, something like protein is a better thing to compare with. Okay. Now, I have a little bit of math, just a little bit of math. It's still before lunch, I'm allowed a little bit of math, right? I want to talk about the BLOSSOM matrix. BLOSSOM. Now, I was graduating high school when BLOSSOM was published, so it's been a bit of time back there. But Hennekoff and Hennekoff came to this notion that we need to figure out which sequence letters can be substituted for other sequence letters more often than we would expect them by random chance. So they began with these blocks, a sequence database actually called blocks. They took regions of sequences that aligned very, very well with no gaps in them across distant relatives. When I talked about yeast and humans, you could even find some blocks that had both human and yeast sequences in them. That's the kind of evolutionary distance we're talking about. And then within that, they said, how frequently do we see that, for example, a leucine was replaced by a valine? How frequently was a leucine replaced with a proline, or a leucine by a tryptophan? So of those three possibilities, leucine getting replaced by tryptophan, proline, or valine, which of those seems like the likeliest event to you? Do you guys know the structures of the amino acids? Oh! I'm grieving for my field. That's okay. I need a toast to the amino acid structures. All right. All right. So leucine is a greasy. It's a greasy amino acid. It's a pretty hydrophobic, just a branched alkyl chain. Valine is like the two fingers, the victory sign for its side chain. Again, branched alkyl chains. Proline is funky. Proline is an oddball. It's a cyclic where the cycle coming off of its side chain loops around to its own N-terminus. It's actually an amino acid, not an amino acid. Bit of trivia for you. And then tryptophan is bizarre. It's like, the giant polycyclic side chain. So of those, leucine getting replaced by valine, just by chemistry, makes sense. But what does evolution have to say about it? Logarithms, log odds ratios, they're a great concept to know about. In short, we can say how frequently do we observe in this blocks database one letter gets replaced for another versus what we would expect. Different amino acids have different expected rates of use because they are used less in in protein sequences. Leucine, for example, is the most common amino acid. Something like 10 or 11 or 12% of all amino acids in the database are leucine. Tryptophan, on the other hand, is extraordinarily rare, and one of the reasons is it's costly to manufacture for our cells. So you would not expect to see W getting randomly appearing than you would versus another amino acid that's much more common. So we can use the expected amount that we would see an amino acid replacing another versus the fraction that we actually see in these blocks. Okay, that by itself is probably a little complex, but BLOSSOM is extraordinarily useful, and it shows up everywhere. So BLOSSOM is definitely something to know about. So BLOSSOM 62 is one of the most common sequence substitution matrices that we use. The term substitution matrix is definitely something I would know, because every aligner needs to have some way to weight one letter getting replaced by another. Okay? So along this diagonal, we have very large numbers. Those very large numbers tell us that this letter gets replaced by itself. Okay? So that's not much of a replacement. If M stays M, that's not really big news. But we have to have a score for it, and that's what these numbers represent. This is a log of odds ratio table. When you see a 0, I want you to think this substitution happens no more and no less frequently than I would expect by random chance itself. Okay. Now I've highlighted in yellow on this slide a whole bunch of letter-for-letter combinations that happen more frequently than we would expect by random chance. Now I said a moment ago that leucine replaced by valine is something that seems biologically quite plausible because they're both branched alkyl chains. So in this matrix, we can look at the L column right here and look for V. Where's V fall? All the way down here at the bottom. Okay, and we see that it has a yellow number. It's a positive number. Now if we flip back one slide, we see that this is a log base 2 matrix, which means that when we see that we see valine replacing leucine about twice as often as we would expect by random chance alone. That's how you interpret these. So a log ratio of 0 means the thing happens just as much as you would expect by random chance. A log base 2 of 1 tells us it happens twice as often. A log base 2 of 2 tells us that it happens 4 times as often as you would expect by random chance. Do you have some 2s on here? I see one. All the way in the second column. Arginine gets replaced by lysine about 4 times as often as we would expect by random chance alone. What do arginine and lysine have in common?
Basic amino acids.
Oh, bless you. Yes. Thank you. Thank you so much. They are both basic amino acids. That's brilliant. So we see in the scope of evolution that replacing an amino acid with something that has a similar functional property is more likely to be viable. Now, I learned just yesterday of a tuberculosis gene that if you change a serine into a threonine, it becomes highly drug resistant. Isn't that wild? Now, serine and threonine are very, very similar. They both have alcohol side chains. Threonine is just 1 alkyl longer. But that single change makes this gene almost completely resistant to the drug we would use to treat tuberculosis. Wild. Okay, so when you are looking at one sequence versus another, a substitution matrix is your best friend. Now let's talk about how that plays out. It's the same matrix down below, but I have at the top one sequence and a second sequence. I've got the Regad sequence and the YDG sequence. And I want to say, how do I score the similarity of these two sequences? So I look at the first pair, R versus Y. R is the second column here. Y is the next to last column. It's a -2. So I score that as -2. I've got E versus D. Now people should recognize there's some chemical similarities between E and D, right? They're both acids. They're both acidic side chains. So I can go to the E column. Right, here's E, and scroll down to D, it's the 4th row, and I see it has a value of 2, +2. So for E versus D, we add 2 to our score. So you should be able to see where all of those values in the second row come from. They're just lookups of one residue versus the other. The score for the alignment between these two sequences is the sum across all of those values. Clever, huh? All right, so substitution matrices give us the ability to evaluate the similarity of two sequences based on known homologies found in the databases as of 1992, and they haven't changed a whole lot since then. So when you're scoring an exact similarity like L versus L, you get a pretty good contribution there. We see that was a +4. Methionine, however, gets an even bigger bonus, a +5 for M staying M. The reason being that methionine is a much rarer amino acid. So that's a great thing. You'd actually see a pretty huge score if you have something like a tryptophan remaining a tryptophan. That's a +11. It's a huge bonus on the log score. Okay, so this is the scorecard. This is how we evaluate whether two sequences are similar or not. BLAST is kind of the amazing success of the bioinformatics field. The fact that this algorithm is still in use since its 1990 publication. It's racked up more than 127,000 citations across, I think, 2 different manuscripts that explain how it worked. I want you to understand how BLAST works. So I'm going to show it to you in text first, and then we're going to look at it graphically. So first off, the authors of BLAST thought to themselves, I can really speed up this whole alignment process if I assume that a long region of similar sequence will contain some small regions of exact duplication. You see that? If two sequences are similar to each other, I can assume that some part of their information is identical. So if I happen to be a science fiction and comic book fan at the movies. Anyone else in the room?
Yeah.
All right, there we are. So we have multiple fans of this class of movie in the room. If someone is marketing to me and they want to find other potential customers, they might ask, did you see Civil War, the Captain America Civil War movie? Good, okay, we got that in common. Have you seen Aliens? Not Aliens? Oh, it's a scary movie. It's a lot of fun though. It's a lot of fun. Classic for a good reason. That's fine. Have you seen?
I've seen it.
Well, I haven't seen Spider-Man. I don't know if I can put that one on the list. But what about Batman versus Superman? Yeah, okay, that's two.
The Ultimate Edition.
All right, awesome. So what I'm saying is that even though he and I haven't seen identical sets of movies, we have seen overlapping sets of movies. And I'm sure we could find at least 3 movies that we have in common. In the same way, BLAST, in trying to search a database for a sequence similar to a query sequence, the one that you give it to look for, will assume that at least 3 letters of amino acid sequence are exactly the same between these 2 sequences. Okay? So for its first attempt to find potential matches against a query sequence, it's going to look for any 3 letters in the original sequence that are exactly matched by any sequence in the database. And only those will be considered for sequence alignment. Okay? So once it has found these seed matches, these regions of identity between the 2 sequences, it will try to extend them to walk out from that region of shared seed match to find letters nearby that are similar in terms of the BLOSSOM score. Once it has done that, it can sort all of these maximal segment pairs, these extended regions, by score, and report those as the hits that we see for a particular sequence. But there's some real magic in BLAST that's really worth talking about, and it was actually published in this Carlin and Altschul paper in connection with it. You see that this was published the same year, different journals though. That is the expectation value. And again, expectation values show up everywhere in bioinformatics. Definitely worth your knowing how to interpret these. So if you get back the top match from BLAST, if it just says this is the top match, that's not so useful to you. You would like it to have some way to evaluate whether that match is something that happened randomly or happened because of the particular properties of the sequence. So how many different alignments with scores equivalent to or better than the score S? S is the score you saw for your best match. So what is the number of different alignments with scores equal to or better than S that are expected to occur in a database search by random chance? The lower the e-value, the more significant the score. So if you run BLAST and your top score is a 1 by expectation value, don't get too excited about it. What that says is, if you had done this search against random sequences, we would expect the best score to look exactly like this, against random sequence. So that's not high. You want your expectation value to be low, not high. Okay, so that bit of math got Carlin and Altschul in a separate paper, but it is also a concept that we use all over the place in bioinformatics today. So, graphically, let us imagine that we are looking for the FIR sequence in our database, and we're currently comparing to RLV, etc. What we see is that there are seed matches, regions of identity, that stretch through 3 amino acids. Oop, just spilled. It was empty, it was empty. All right, so we've got RPE over at the left end, We have MCT in the middle. Both of those are regions of identity that stretch through 3 amino acids. Therefore, we can proceed to do this alignment to try to find regions adjoining those seed matches through which we can extend to boost our score. So when we look at A versus V, we're pretty happy. Those are both kind of hydrophobic. We've got E versus D. We already said that one's happy. Those are both carboxylic acids. A versus V. All of these have positive scores on the Blossom matrix. But as we extend out this way, let's see what we have. We have S versus A. Yeah, we'll take it. We have F versus F. Very happy about that one. Phenylalanine is a massive amino acid. It's pretty rare, and so replacing this amino acid with itself is very happy. K versus H, well, that's okay. Histidine is kind of a basic amino acid. D versus E, we already said it was great. N versus N, no problem. Very good there. But what about this P versus E business? How often is proline and glutamic acid, how often are those able to carry out the same function in a protein? Not so often. And when we look at that replacement on a Blossom matrix, we see a low score. So at some point, we can no longer extend out from these seed matches because we run into regions of sequence that are just kind of appalling in their similarity. And at that point, we look for building the score as highly as we can, and after we've taken a few negative steps to diminish that score, we say, okay, we're not going to find a better alignment than this short one that we have. So that's this process by which we start with a seed match and extend out to a maximal segment pair, which refers to this the wider region built from a seed match that maximizes our score. You have a question?
So does the algorithm just look at one seed match initially, and then so it doesn't need more than that?
That's actually the reason why there was a second BLAST paper. So the first BLAST paper relied on the presence of only one seed match. But the BLAST servers that we use today all run the new code, which requires to have 2 seed matches occurring within a protein sequence. So it's actually given, that change makes it even less sensitive to very distant homologs, but it certainly increases the speed tremendously. Good question. Other questions? Okay, that's BLAST. Everyone should use BLAST at least once or twice in your life. It's great software. All right, now, let us imagine that there's no long homolog for a particular query sequence. We have we've looked for whole sequence matches and we're just not turning them up. And we still want to know, what does this protein that we've just discovered in organism X, how do we determine what it does? One of our best options is to look for sequence motifs and structural domains. So at the top, I've written something that looks a little bit like the central dogma of structural biology. Have you all heard this one?
Yeah.
We all hear central dogma for molecular biology in general, but the central dogma for structural biology is a little bit different. We have sequences. Those sequences take on structures, and those structures in turn allow a particular functionality for that bit of sequence. So I think of energy minimization as this process that helps us infer what structure a sequence takes on, and then usually by affinity or catalysis we see that structure implies a function. So imagine then that we have sequence motifs that we've recognized that are characteristic of a particular biological activity. So it's a conserved element where the protein sequences all bear similarity when they carry out this particular kind of functionality. Similarly, we can see that it matters less what sequence you have at a particular site as it does what structure you take on in order to carry out some certain function. So we may want to look in the sequence domain to try to make comparisons among sequences, or we may try to infer this on the basis of structural similarity. And of course, as we've mentioned in orthology and in parology, we see that protein families, whole sets of protein sequences may bear similarity to each other. Okay, so the tool, there are several tools that we're going to discuss, but one of the most commonly seen ones that we talk about is called a position-specific scoring matrix. Now, if you want to be hip, don't call it a PSSM, because that's just too long. call it a possum. Now, you don't have possums so much around South Africa, but where I grew up, you see plenty of possums. Generally speaking, if you run out in the middle of the night to put something in your dumpster in the middle of the night, and something makes a noise and scares you, it's a possum that's been rooting through your trash, right? No fun. Okay, so possums. So if you have a sequence motif, a possum is one of the ways that you're most likely to represent it. So each column of a possum represents one letter of sequence. So you can think of this sort of like a hidden Markov model where each state is a subsequent letter in a sequence. I'm not going to worry about that too much, but basically, if you can align a whole bunch of proteins, maybe 20 proteins, let's say, that all have the same kind of functionality, you'll be able to say what letter does each of these sequences have at this particular position. And from that possum, you can infer a pattern that you use to search for it. All right. Now, there are lots of ways that people employ possums in this modeling. Interpro is one of these very useful sites. I'm coming up on the end. I don't think we're going to run too much longer here. Interpro is this great resource out there for allowing you to look for particular sequence features that have been documented by all kinds of different fingerprint makers around the world. So you might use regular expressions. This is a computer science term that you see a lot among people who use Linux. They just call them regexes rather than regular expressions. You know, it's good to have some hip lingo for your subfield. So you might use regexes as a way to recognize these sequence similarities. So for example, a regex might use something like square brackets and say STY, for example. So here I've got a square bracket, here I've got a square bracket, STY. What that means is at this position, any of these three residues is acceptable, ST or Y. Okay, so that's an example of a regex expression. You might use a formal possum to develop a statistical model about which sequences out there are best aligned to a possum. Prince is a very good example of a database like that. Or you might see automated clustering used to find sequences that are most similar. So something like ProDOM uses that. Now, hidden Markov models are hugely popular for this topic. PFAMs, Smart TigerFAMs, all these different systems make use of hidden Markov models here as a way to recognize complex features that might be present. So like transmembrane domains or something like that. Maybe you want to model that with a hidden Markov model. So InterProScan is very closely tied to UniProt KB. UniProt is one of the biggest databases of protein sequences out there. People use it for all kinds of purposes. Now, I want to point out that Nikki Mulder, who runs the CBio computational biology group over at UCT, was one of the authors behind Interpro. So if you have interesting questions about that topic, you might want to chat with her about her work with the project. But Interpro is still very much under operation. You can even download this to run it on your Linux server to process whole, thousands of sequences at the same time for little bits of functionality within those proteins. I actually have a server set up for doing this, so I know. Now, in my checkered career, I have served as an expert witness for federal trials in the United States. And I worked to defend people who had been accused of manufacturing ricin. So think less of me if you will, but as a result, I've gotten to know ricin pretty well. You would be appalled if you knew what the government used to use to prove people had produced ricin. It was not very viable. It's okay. I think they've learned their lesson. That's fine. So, ricin is a very dangerous protein. What happens if you swill a cup of ricin? If you swill a cup of ricin, you are very, very dead. Very deadly. In fact, there have been some Russian emigres who've been accused of being spies by their government, who've been killed by people using ricin. A fellow manufactured a special umbrella with a little needle in the spike, and in a crowded subway station, he poked a Russian emigre with his umbrella and injected a tiny amount of ricin into the guy's body, and he died horribly. Horribly. Terrible way to go. So, why is ricin so dangerous? Ricin has two different regions to it. Part of it, near the C-terminus, is a lectin domain. Does everyone know what a lectin is? Lectin. Have you heard the term? Oh, okay. All right. Well, a lectin is a region of a protein that is able to recognize sugars. Sugars. So if you have a sugar on a cell surface, a lectin may cause you to recognize that sugar. And then on the other end, it has a ribosome inactivating protein. Now, just to give you a clue, these sites frequently abbreviate that section as RIP. What do you need ribosomes for?
Translation.
Absolutely, for translation. If your cells can't translate proteins, you are dead. So you can think of ricin then as being, as recognizing sugars on cell surface. The sugar in this case, ricin in circulation will attach itself to a sugar on a cell surface, and that attaching to a sugar causes the cell to take that protein into itself. All right? So think of this as a door knocker, right? Hello, Mr. Cell. I'm a protein you need to let in. It lets it in. Immediately, it starts killing ribosomes in that cell. So that's the ricin protein. You can see that we've got matches from all these different engines, like the ProSite, like SuperFam, PFAM, all of these different sites. All of those fingerprints are available under one umbrella. If you pass a sequence through InterProScan, it's going to use all of those scanners to look for these features and give you back one pretty report where you can click on any of these to learn information about, say, the 3D structure that caused this region to be recognized, or the how is it that the signature for the lectin was recognized by SM, for example, by the smart links. So we have all of these different tools, all made much more accessible because it's all piled within this. In fact, any protein in UniProt has already had this done for you. You don't have to search it again in InterProScan because that result is already there for you to go poke at. So this is a very powerful site, one that a lot of people should use. A lot of times when you're just looking at a big FASTA database for a species, it'll say unknown protein or hypothetical protein. And yet you can still come to the InterPro result for it and see that actually we know a fair bit about this. We can say, this protein is DNA interacting. It's got a leucine zipper right there. So that's very powerful. Keep InterPro in mind when you want to know, what does this thing do? All right, so we've covered a lot of material, I appreciate. But I hope that a lot of this has given you clues as to how we deal with novel genome information, how we decide, how we get from those basic sequences to contigs, to whole chromosomal assemblies, how we've dealt with dealing with screening out repeats, for example, how we decide what functions those proteins take on, where the genes are to be found in a long sequence, for that matter. So all of that I've tried to sum up in one page here, but I hope that you find that this lecture is useful in deciding which bits of sequencing end up where, and how we decide where the meaningful features within a genome really are. Now, are there any questions before we go? Okay, so I'm going to see you guys 3 times, this week, next week, and in 2 weeks. Each time I'm going to be talking about the bioinformatics surrounding the biology and the technology that Bronwyn's been talking to you about already, so I hope you find this useful. I will post this video to my YouTube channel, and I hope we'll be able to get that link sent out to you guys so that you'll have copies of the slides and of the video. And I try to spend each Thursday over at this campus. Usually I'm sitting over by the mass spec people though. So if you have questions, I hope you'll come by and say hello. All right?
Thank you.
Great. Thank you. Thank you.